It is really a pleasure to welcome Professor Colman. She's from Argentina originally, but she had obtained her PhD actually in Spain at the Institute of Biomedical Science. And then she's moving now at the University of the Medical Center in Utrecht as an associate professor at the Center for Molecular Medicine, where she is leading a group that investigates the metabolism of stem cells and cancer at the subcellular, cellular, and tissue scale. And in her lab, as a model system, they use organoids models that can recapitulate the complexity and differentiation dynamics. And her lab also performs cutting-edge advanced 4D live imaging to gain insight of metabolic changes, temporal resolution, using in particular also fluorescent-based metabolism, and cell type specific changes.
Thank you so much for the invitation. I really look forward to share our research with you, and hopefully we can have some nice discussion. Thank you for the nice introduction as well. As it was mentioned, our lab is now in the Utrecht in the Netherlands, and we have a passion for metabolism and how metabolism could regulate the cell fate decisions. Yeah, so that's a normally conventional way of understanding metabolism and cell fate. And when I say cell fate, I mean as if a cell is going to differentiate into a specific cell type, or is going to go out from the cycling cycle, become quiescent or senescent. The conventional view is that these cell fate determinations would have an effect on metabolism, meaning that the cell changes and consequently the metabolism of the cell changes. But we are more interested to investigate whether metabolic changes that could come from the environment could be regulatory on cell fate decisions. And the way we look at that, we consider that this link could be taken by cell signaling. Let's say, for instance, that metabolism could affect the redox signaling, but also modifications of proteins. And another way that we are interested in investigating is how metabolic changes could impact in enzymatic activity, specifically of epigenetic regulators, and in that way rewire transcription and then change the fate of the cells. And as I mentioned, we call cell fate to processes such as stem cell differentiation, but also plasticity, but also what happens, of course, in cancer when the tumor develops and upon chemotherapy response. One of our favorite tissues, organs, is the intestine, and that's basically because it puts together two of our most interesting questions, which is metabolites. At the very beginning, they come from food, right, from digestion, and the intestine and the epithelial layer, where when these metabolites will be absorbed. Also, it's interesting to us to study the intestine because in this idea of understanding cell fate decisions, the intestine, the epithelial layer, which would be this one, is made out of specific cell types, stem cells that they cycle all the time, and they actually reconstitute the whole epithelium. But they also give rise to specific cell types with functions, for instance, enterocytes and secretory cells. Enterocytes will be the ones that we locate up here and will be in charge of the uptake of nutrients, while secretory cells, there are different kinds, and they are more in charge of protecting the epithelium from, for instance, all the bacteria that will be present. Previous research of myself as a postdoc, but also from the lab, has shown that if we actually look at these different cell types, we will find that they have different metabolic profiles. We actually find changes in the amount of mitochondria that these cells do, and also the way they make energy, the way they make ATP. Further studies from our lab and also from others have shown that in this process of differentiation, where these stem cells have more mitochondria and the secretory cells have less mitochondria. If we alter the mitochondrial state on these cells, that can be sufficient to make these cells to become a differentiated cell. And one of the other aspects that is also relevant for the talk of today is that we also found metabolic interactions between these two cell types. Do you see my mouse or? Yeah, okay. So that we find a metabolic interaction between these cell types. Specifically, we saw that lactate could be exchanged from differentiated cells and be taken up by stem cells that will use it to make energy. With these interesting observations, now we wonder what happens in cancer, what happens in the tumors. If we think about the tumors, they arise as a consequential or sequential acquisition of different mutations in all those pathways, for instance, Wnt pathway and RAS pathway and P53, but these pathways are normally in charge of make these cells either stems to regulate the process of stem cell differentiation. So when we had this mutation, it was normally acknowledged that then the tumor will be made of a bag of proliferative cells that have lost the capacity to become differentiated. So a bag of cancer stem cells, if you would think so. But actually, that dogma has been challenged not that long ago, because what it has been shown that even in the presence of these mutations that, let's say, will make all these cells to be behaving as a high Wnt cell, they will still show some differences in their activation of pathways. If we look at Wnt pathway, which is the key pathway that regulates proliferation and stem cell features, they actually found that the tumors will have, the red here means the Wnt signaling. They will be all high in Wnt. And if they will actually remove these cells that were high in Wnt, for a moment, the tumor will stop growing. But following up on time, they will see that these cells will regain the capacity of become cancer stem cells, high Wnt cells. So this showed two things. One, that tumors are heterogeneous because they can have this differential Wnt activation with cancer stem cells, cancer differentiated cells. But they also showed that this will be very plastic because cells that are cancer stem cells can go into differentiation, but also the other way around. And later on, it was also shown that this is functional because, in other ways, for instance, in metastasis, because the existence of these two types of cancer cells, differentiated and stem cells, was required to successfully outgrow metastasis in the liver. So that's a bit about the tumor biology, at least in the colon, the colorectal tumors. Now, how does this connect to metabolism? So again, the conventional, one of the most conserved metabolic phenotypes of cancer cells is the Warburg effect, where cells will enhance their uptake of glucose and they will actually use glycolysis with an end product of lactate, even in the presence of oxygen. And that has been accepted and long seen by many in different cancer tumor models. So that we all agree on that, and even in the colorectal cancer, if you look at the levels of glucose, they're indeed decreased in the tumor microenvironment, and also the levels of lactate are increased. So we wanted to look at this heterogeneity also from the metabolic perspective. So one of the first things we did was to make a simple staining of the monocarboxylate transporter 4, which normally people consider to be higher and more glycolytic cells. And if we look at the tumor, here, you would see that the staining is not completely even in all the cells. And if we at the same time look at a marker of differentiated cancer cells, we also see it's also not even. There are some differentiated cells, and it's a nice correlation between these two. So this was a first very basic observation that make us wonder whether there will also be metabolic heterogeneity in the colorectal tumors. To be able to go a bit bigger and to address many tumors, we were able to find data sets where single-cell transcriptomics was done, and take information from 30 tumors that came from 2 different studies. And then what we did is to reanalyze the single-cell RNA sequencing and try to find different metabolic signatures. So first of all, we could observe that within these tumors, we will always find some cell types signatures that were the ones described in the healthy tissue. So this has also been shown by others. It seems that these colorectal tumors, although they are high-wind and they're more proliferative and they lose all sense of architecture of the normal tissue, they still have tried to recapitulate this differentiation that is observed in the healthy tissue. So if we look now at these signatures, like the glycolytic signature and the OXPHOS signature, we do see some differences. Interestingly, we see that these cells, which are differentiated in the tumor, seem to have higher glycolysis, while cells that are proliferative and high-wind, these cancer stem cells, seem to have higher mitochondrial activity. Of course, here I don't need to mention for this audience that looking at transcription to try to guess metabolism is not the right way to go. I mean, it could be indicative, but it's of course not sufficient. So we need to move forward to try to understand metabolism in a better way. So these differences that we find, are they regulatory? So then we find that these metabolic differences may have an effect on tumor biology. To try to address that, we need to be able to simultaneously look at metabolism, but at the same time to look at these cell fate decisions. And we also need to have some single cell resolution because otherwise we cannot look at the heterogeneity. So because of that, we decided to work with human tumor organoids. I hope you are familiar with this, but basically we can take from the colon in this case, either healthy cells and make them grow with recapitulating the tissue architecture in vitro. We can also take from the tumor and we can do something alike. So this is a very nice model because it conserves many of the characteristics of the tissue of origin. So in these organoids from patients, now we can introduce specific genetic reporters. These genetic reporters are designed to either follow Wnt signaling to have a readout for cancer stemness, but we also combine them now with reporters that can measure either glucose or pyruvate or lactate or the NAD-NADH ratio flux. And this is a bit how they will look like. So now we have in the same cells, we can have two readouts, is the cell a cancer stem cell or not, and what's the metabolic state of the cell. Here's one of the examples to measure lactate. We use this laconic reporter developed by this lab already some time ago. And the rationale of these reporters is that we use are either FRET-based or radiometric. In this case, this FRET reporter has two fluorescent proteins, and in the middle of them, they have a binding pocket for lactate. So the binding of the lactate will change the conformation of the protein, and this will reflect in the emission and excitation parameters. So here you can see, for instance, if we just inject lactate and if we image the FRET ratio, we could see an increase in the signal. With that type of reporter, we now just make pictures of our organoids where we can have the star probe, which is the Wnt reporter, high Wnt and low Wnt. We now look at the ratio of FRET, and we actually found a nice difference where these cancer differentiated cells, so the Wnt low, so not the cancer stem cells, but the differentiated ones, have higher levels of lactate. Now we can exploit this further because making pictures we could also do with different ways. So now I want to use the reporters to actually exploit also the time resolution. Now we can actually monitor whether they will respond and which kinetics will they show. So one of the experiments we did was to decrease the amount of glucose and actually evaluate whether lactate could be taken up by the cells. So this is what we did here. And you would see, then you see the increase in FRET, and that actually corresponds and the increase of lactate in the cells. And we can, of course, quantify this. And the other thing that is also interesting to us is that we also have a reporter for the cancer stem cell state. So now we can actually deconvolute this response and look whether different cell types, we have a different way to uptake lactate. And that was indeed the case. And if you can see here, the black line, this corresponds to a higher lactate uptake from cancer stem cells. So now you put this together, we have cells that are higher in lactate, which are the cancer differentiated cells, while the stem cells can uptake more lactate, which actually supports a notion where this lactate could also be shared within the tumor cells. Then we wonder whether if this happens, and we can come back to the idea that the lactate, the environment of the tumor is facing all the time high lactate concentration, could it have any functional role in the tumor and the development of a tumor? Many things have been said in this direction. Actually, lactate has been able to promote metastatic behavior, but also to avoid immune cells to reach the tumor. But whether lactate could have an intrinsic effect on tumor development is something that was a bit unclear yet. So we said, okay, let's challenge our organoids, tumor organoids with lactate and see what happens. So we look at the stem cell state because we observed that the cancer cells were good at taking up lactate, and we actually found that only increasing the lactate concentration in the medium would increase the number of cancer stem cells. That was not only by looking at the images, but we can also quantify this by facts. And we also look at transcription, and we actually saw a very nice increase in the cancer stem cell genes, while the genes that report for differentiation were downregulated. So let's go a bit more fun into the imaging again. That's interesting to know, but that doesn't really resolve how lactate made more cancer stem cells. If we look at the conventional way of what happens in tissues, you have a population of cells, of the cancer stem cells, or let's say healthy cells, and these cells will differentiate. So you could also postulate that in the tumors, what happens is that lactate is inhibiting the process of differentiation of the cancer stem cells. Alternatively, we could speculate that maybe cells that are non-cancer stem cells, so differentiated cells, could fall back into a cancer stem cell state. With the conventional ways, if you try, well, there is no time here, but if you try to picture how would you look at that, yeah, it's difficult. So we need to apply imaging again. And here you can I will share with you this long time lapses where we can actually follow metabolic state and the stem cell state, and we can also see them merge. If we only look at this and try to wonder what's going on, it's a bit difficult, right? If we want to apply conventional tools to address this, it's also difficult because these nuclei, they don't have an homogeneous shape, and there is a lot of movement in the C-stack, and it's also, of course, a matter of time. So we applied a machine learning-based method in collaboration with our collaborators in Amsterdam, and now we can train a model to try to predict where the nucleus are, and when the cell divides, which are the daughters. This was very, we invested a lot into this. It was a lot time consuming and effort consuming, but actually turned out to work quite okay. So now we can convert this information into vectors, and we can digitalize this information. And here you can see now the representation of the growth of the tumor. The first different colors will tell which cells are from the same family. The middle panel is the stem cell state, and the latter panel are the changes in the NAD/NADH ratio. So we can now nicely reconstruct how this tumor came to be. Now we look again at our effect of lactate, going back to the question, and then here we are plotting the levels of the cancer stem cell state. You can see that in the control there are some cells remaining, but they tend to differentiate. But in the lactate, we see many more red popping up. So this is only a fraction, a portion of the reconstruction of the tree that we can make, because as I said, we can capture every single cell. And here you can see every time this branches, this means it's a cell division. So if we, for instance, look at these two, you can of course see more red and more green. And a nice way to look at these trees is, for instance, there's a lot of information that we can gain for here. For instance, the distance between two branches will now represent the cell cycle length. So we can measure for each of the cells how long it took for them to cycle again. We can also count how many cells from how many clones happen. So from one single cell, how many times did it divide? We can also look at the symmetries. Here, for instance, in the control, you can see that these two daughter cells, they enter the next division round kind of symmetrically. And this happens quite robustly. Even in the tumors, we observe that this happens. So every time the cell divides, also the other cell will divide, even if they're not closer in the space anymore. Now, if we look at lactate, things look quite differently. First, we found a much higher cell cycle lengths. So the cell cycle length was much longer. But when we look at the organoids, when culturing them, we actually did not observe a difference in how big they were when we compared the two conditions. So how could this be explained? So what we also assessed is how many cells were cycling, or at least how big were the clones. So one, for instance, means that a cell that has not divided at all, so the clone size is 1, this is a cell that actually gave rise to 2 daughters, so clone size 2, and here you have, for instance, clone size 5. What we observe now is that in the case of lactate, the amount of non-cycling cells reduces, while the amount of cells that give higher clones increases. And that helped us to understand that actually what was going on is that although cells were cycling slower, now we know that most cells are cycling upon lactate. So very fundamental changes in the behavior of these tumors just by facing lactate in the environment. Now, going back to the question, do we see cells looking differentiation or the differentiation? We actually found, well, in the control, 35% of the cells will differentiate, so they will leave the cancer stem cell state, and only a small portion of cells will come back to a cancer stem cell state where they cycle. When you put them with lactate, we see a hard reduction of differentiation. So differentiation is inhibited. But more impressive or more striking to us, there's a huge increase on the differentiation. So now lactate, what it's doing is those cells that were not going to cycle anymore, that were part of the tumor, but they were differentiated, they are now coming back into a cancer stem cell state. So it's a bit of the summary. There is much more that we did with this analysis. Also, we look at the neighborhoods, and this is still work in progress, and we are very, very happy with this development. If we think about mechanism, what we did was to see in which ways lactate was altering the metabolism of the cells. And we found actually nice increase in the TCA cycle intermediates, and specifically high levels of acetyl-CoA and pyruvate. So we wonder whether this could come from lactate being converted into pyruvate and acetyl-CoA, so that will fit, because we know this happens, of course. With that in mind, that means if the effect of lactate would be driven by pyruvate and acetyl-CoA, if we will just put pyruvate instead of lactate, we should see the same effect, and that's indeed the case. Higher levels of pyruvate also increase the cancer stem cell population, and if we inhibit the production of lactate in the cells, that will also lead to increased pyruvate. It will also increase the number of cancer stem cells. We also try inhibition of PDK, that will also increase pyruvate, basically acetyl-CoA, and we also see an increase on stem cells, and that's depending on activity of mitochondria. However, we did not find a difference if we will block pyruvate entering mitochondria, which makes us wonder what's going on. This is a bit more into very deep cellular metabolism, but long story short, 2 things. First of all, lactate can enter mitochondria directly and be used as a substrate for the transport chain for oxidation, basically. And on the other hand, we also find that pyruvate and actually acetyl-CoA could be relevant somewhere else and not in mitochondria. So we did RNA sequencing of these conditions, and we find a nice list of genes. Many of them had to do with the Wnt pathway, as we already expected. But we also find some categories that were a bit unexpected, like nucleosome assembly and chromatin organization. So for this audience, maybe I don't need to mention this, but just in case, you know now that metabolites can be substrates, but also cofactors of many of the metabolic epigenetic remodelers, meaning that they can have a direct impact in the chromatin conformation and consequently into gene transcription. We actually observed that upon lactate, the levels of acetylation of histones will increase. So how that affect the accessibility to the genome? Well, we can do ATAC sequencing, which actually is exactly that. It will tell whether the chromatin is more accessible for transcription or less. But of course, we can also now see where in the genome that will happen. So if we go back and take the genes that we find significantly upregulated, we now can we can integrate these two data sets. Now we find that when we took a random group of genes, this is how much they are open in the genome. But if now we take the group of genes that were upregulated on lactate, we can see that the distribution is different. And here in this tail, we find those genes that were more upregulated upon lactate, but also where the chromatin is more open because of lactate. And among these genes, which will be epigenetically regulated by lactate, we find MYC and others. But I want to make this story a bit simpler. But actually, we do think it's about MYC. And if we go back to the patient material and look at a single cell RNA-seq, we do find indeed that MYC is expressed in those cells that are having high activity of mitochondria and are cycling. Here is the signature here. While the cells that are differentiated are really glycolytic. So if we can now go back to the organoid models, and then now we see, okay, what will happen if we now put lactate again, but now we don't allow the effect of MYC and be managed by the inhibition of BRD enzymes, which actually read the acetylome and won't allow MYC to be transcribed. If we quantify back, this is I showed this already. So we have a decrease in differentiation when lactate and increase in dedifferentiation. If we look at the effect of inhibiting MYC at the epigenetic level, now we find that differentiation is partially restored, but only partially, while the effect of the differentiation is strongly reversed. And this made us think that the effect of lactate on epigenetics will regulate the differentiation, so plasticity of the cells, while the effect of inhibition of differentiation is probably regulated by other means. With that, we come to this model that shows that we have in the tumor, we do have the cells trying to recapitulate some of the features that are found in the healthy intestine. And we find lactate, which is an abundant metabolite in the tumor microenvironment, having a very active effect in the regulation of the cancer stem cell state by altering many of the features of the tumor development, like cell cycle length, clone size, and overall Wnt signaling. So that is one story that is we are now rebutting. We're happy about it, but well, still very busy with it. But I wanted to bring another story, a short one, I hope, where I want to highlight what's the importance of looking metabolic reporters to be able to have high temporal resolution and even subcellular resolution. So here we'll go a bit faster because I want you just that to be the take-home message. So if we think about the cell cycle, we all know G1, where cells grow, S phase, where cells duplicate, G2, and mitosis. Mitosis is a very drastic cell cycle phase where many things happen, but it's also very short in time. It takes, depending on the cell type, from 30 minutes to an hour, and within that, the whole DNA has been divided between the two cells, but also the organelles. And a lot has been, not a lot, but yeah, studies in this matter in the topic of metabolism have focused on how much ATP, how ATP is important to support these processes by all the energy it takes to move things around. But beyond ATP, not much is understood. Another aspect is that we can use our metabolic reporters to try to understand a bit better how metabolism changes in mitosis and where it could have a regulatory role in mitosis. So these reporters that do not only give us resolution in terms of time, but they also give us resolution where in the cell. We can put them in different compartments and have subcellular resolution of metabolism. Here, for instance, we put a reporter to measure ATP in mitochondria, and she can see here that we do find a nice and sharp, a nice peak, a nice increase in ATP in mitochondria when cells go into mitosis. We can also put reporters fused to H2B. If we put them fused to this histone, we can actually monitor metabolic changes on the chromatin. So we look here, for instance, at the levels of NAD, NADH in chromatin by fusing H2B. And now we find, this is not mitochondria anymore, this is on the chromatin, a double peak that will appear increases of NAD when cells, after the nuclear envelope breaks, when cells enter mitosis. So one of the questions was, what is the origin of NAD/NADH? Would be that be glycolysis or would that be mitochondria? And I think you may know that the last step of conversion pyruvate to lactate does give back oxidized NAD. And in the mitochondria, there is a lot of NAD production. Is NAD oxidation from NADH in the transport chain, and that can connect through the cytosol through different shuttles. So that was the question. This peak, where does it come from? So if we want to evaluate if it comes here from pyruvate to lactate, we can actually measure the levels of lactate in mitosis. And this becomes now more intriguing because when we look at the levels of lactate in the cytosol, we can see here the different mitotic moments. We actually found a drop indicating that there will be less lactate at least produced or less lactate in the cell. Now, because our phenotype of NAD/NADH was on the chromatin, we also look at lactate levels in the nucleus. And here, what you can find actually is not a decrease like in cytosol, but an increase. So different behavior in different compartments. So we did metabolomics. If we do metabolomics, actually, we find no differences in cells that are in G2 or cells that are in mitosis. Although the amount of lactate in the media is decreased. So altogether, if we put all together, we actually found that lactate was compartmentalized. So in the cells, the levels of lactate are higher in the cytosol than in the nucleus. But when mitosis occurs, these two compartments fuse, and now they become in one compartment is sensed differently. For cytosol, the levels will drop, while in the nucleus they're going to increase. But overall, we actually found less glycolysis going on in mitosis. So alternatively, we look at mitochondria. When we look at mitochondria, we can inhibit the different transport chain the transport chain components, and then we saw that for all of them, we found a drop in the NAD/NADH on chromatin. This showed that mitochondrial activity is contributing to this peak observed in mitosis, and also the usage of this inhibitor, which inhibits the shuttle from mitochondrial cytosol, also drops the levels of NAD/NADH on chromatin. So it comes from mitochondria. Is it a functional role? Here we can go a bit quickly. To test the idea of being functional, we need to be able to disrupt the peak. But if you use these inhibitors that we talk about, we do know that these inhibitors will also block ATP production. So that won't be a readout, a useful way to assess the effect of NAD/NADH. So alternatively, we actually use lactate, because lactate, by inhibiting this reaction, has a dramatic effect on the levels of NAD, on the ratio NAD/NADH, which is proven here. If we put lactate, the ratio will drop. If we put pyruvate, the ratio will restore. So if we put lactate, now we can see that the peak that we observed before is attenuated. So one of the things we look at was the length of mitosis, and we actually did not find that that addition of interruption of the NAD/NADH will lead to a longer mitosis. But we did find that now all of the sudden cells will start making errors at the time of segregating their genetic material. And how that could be, again, I'm not going to go to it because it's a bit taking too much time, but I would say that there are different ways, and then NAD actually is a cofactor of sirtuins, and sirtuins are important regulators of histone deacetylation. But you need to go from an interphase DNA where the DNA is decondensated to a condensated version of the DNA. You need to deacetylate the histones because that will reduce the charges and will enable the compactation of the DNA. So now we find that this NAD that we saw upregulated in mitosis is actually an important contributor of the process of chromosome condensation. So this is a bit the model. If we have an unperturbed system for this project, we do have a peak of NAD/NADH. Oops, sorry. These levels of NAD are actually important to maintain the activity of sirtuins, which are involved in the deacetylation of residues of histones that will enable the compactation and the proper segregation of chromosomes. Now we can now speculate what would happen in cancer. In cancer, we have, as I already explained even in the first project, we have higher levels of lactate. So these higher levels of lactate now could be affecting the NAD/NADH ratio, as we did with our in vitro system. If that is altered, now sirtuins will be less active, the DNA will not be properly condensated, and now we start to have some errors. Should we care about these errors? Well, maybe. So we have to consider that mitotic errors could lead to apoptosis. And if that happens in a cancer cell, I think we are all happier, because then the tumor will grow less, or tumor cells will die. But there is a risk attached to it, because sometimes cells that carry errors will not go into apoptosis. They will actually remain. And they will remain and become maybe quiescent or not sucking anymore. But sometimes, like here, we had a cell that actually divided, had an error, and divided again. So we now have a new clone with a different genetic makeup that will continue to fuel the tumor growth. And we all know that one of the challenges to efficiently target tumors is this acquired heterogeneity at the genetic level. Then now conventional chemotherapies or personalized therapy fails, because in the tumor we have different genetic populations. So with that, yeah, that was a lot, I think. I hope you guys have any questions. This is the nice lab. Well, we are some more people now, and I think we need to update the picture. But these are the people that were involved in the project that I talk about. And thank you for your attention. And I'm happy to answer any questions.